Hello friends, welcome to SAM Biology class. Friends, today we shall discuss about types of crossing over, factors affecting the frequency of crossing over and its significance. Crossing over is the reciprocal exchange of genetic material between homologous chromosomes. Depending on the type of cell in which it occurs, crossing over is divided into two types. One is somatic crossing over and the other is germinal crossing over. Somatic crossing over takes place during mitosis. Hence, it is also called as mitotic crossing over. It was discovered by Korsten in Drosophila. It also takes place in certain fungi such as Aspergillus. Mitotic crossing over is rare, it has no genetic significance, and it produces patches of crossover tissues between normal cells. Because of this, individuals with mitotic crossing over have mosaic appearance. Mosaics have also been reported in birds and mammals. Somatic crossing over takes place during mitotic prophase. The exact stage in which it occurs is not known. Scientists suggest that it might occur during interphase, whereas others suggest that it takes place during transition from interphase to mitotic prophase. The other type of crossing over is germinal crossing over, which takes place during meiosis. Hence, it is also called as meiotic crossing over. It has genetic significance and takes place in germ cells at the time of gametogenesis. In addition to this, according to the nature of chiasmata, crossing over can also be classified as single crossing over, double crossing over and multiple crossing over. This figure shows the differences between single, double and multiple crossovers. In case of single crossover, only one chiasma is formed along a chromosome pair. So genetic exchange takes place at a single point. But in double crossover, two chiasmata are formed between homologous chromosomes. Because of this, crossing over takes place at two separate points. But in multiple crossovers, several stomata are formed between homologous chromosomes. As a result, genetic exchange takes place at multiple points. Based on the number of chromatids involved in genetic exchange, double crossing over can be divided into different types such as 2 strands double crossing over, 3 strands double crossing over and 4 strands double crossing over. Now let us distinguish between them. This is a pair of homologous chromosomes. These are the chromatids and here these are few genes which are present along the chromosome. In 2 strands double crossing over, only 2 non-sister chromatids are involved in genetic exchange. So 2 chiasmata are formed along the same 2 non-sister chromatids. As a result of this genetic exchange, 2 chromatids become recombinant and remaining 2 remain non-recombinant, thereby preserving parental combinations of genes. But in 3 strands double crossover, out of 4 chromatids, 3 chromatids are involved in genetic exchange. So here also 2 chiasmata are formed, but out of 3 participating chromatids, 1 chromatid is involved in the formation of both chiasmata. As a result of this, 3 chromatids become recombinant and the remaining 1 remain non-recombinant. But in 4 strands double crossing over, all the chromatids are involved in genetic exchange. In this process, out of 4, 2 chromatids are involved in the formation of first chiasma and the remaining 2 are involved in second chiasma. As a result, all chromatids become recombinant. The frequency of crossing over primarily depends upon the distance between linked genes. In addition, several other factors such as temperature, radiation, age, mutagens, chromosomal inversion, nutritional effect, structural changes in chromosomes, centromere effect and chiasmata formation also affect the frequency. First factor is temperature. It has been demonstrated that high and low temperature increase the frequency of crossing over. Similarly, X-ray and other radiation also increase the frequency. It has been demonstrated that in female Drosophila, the frequency of crossing over decreases with increasing age. In addition, presence of certain chemicals such as mutagens do affect the frequency. Gene mutations also influence the frequency of crossing over. Certain mutations increase whereas some other mutations decrease the frequency. In addition, chromosomal inversion suppress the frequency of crossing over. As already mentioned, the frequency of crossing over is primarily dependent upon the distance between linked genes. Greater is the distance between genes, higher is the frequency of crossing over between them. Whereas if the genes are closer, then the chances of crossing over is comparatively less. In addition, concentration of metallic cations such as calcium and magnesium also affect the frequency of crossing over. It has been found that high concentration of calcium in diet suppress the crossing over. Next factor is chromosomal aberration. Structural changes in chromosomes leads to the change in the order of arrangement of genes. Such changes also suppress the frequency of crossing over between genes. In addition, the genes which are located near to the centromere exhibit a lower frequency of crossing over. Finally, formation of chiasma at a point suppress the formation of another chiasma in its vicinity. Now let us discuss about the significance of crossing over. First one is crossing over provides proof regarding the linear arrangement of genes in a chromosome. Second, by obtaining data from crossing over between linked genes, we construct linkage map and chromosome maps. And finally, as crossing over leads to the rearrangement of alleles, it plays an important role in the process of evolution. So friends, this is all about the types of crossing over, factor affecting its frequency and the significance of crossing over.